So biology of B lymphocyte. We've been talking about antigen. We did have a couple of lectures on antibody. And as you know, the antibodies are produced by B cells. So today we'll talk about the biology of B lymphocytes. Like any blood-derived cell, we'll talk about development and differentiation of B lymphocytes. Where are they synthesized? And we will spend a lot of time on the markers and receptors present on B cell membrane and how they are seen by our immune cell and how signaling takes place. So I'm going to introduce one term, signaling, because most of the drugs that you will come across in immunology are going to talk about signaling. So signaling basically means that there is a message from outside and it hits the membrane. Membrane takes up the message, it's processed into the cytoplasm, it goes into the nucleus, and then nucleus starts doing what needs to be done at that appropriate time. So this series of events will be called signaling. So let me introduce a B cell to you. A B cell is a lymphocyte, and one of the most important functions of this lymphocyte is that it synthesizes antibodies. And of course, antibodies will be synthesized only when there is a outside intervention in terms of exposure to antigen. So antigen will come, bind to an antibody, and then this message will be taken up by B cell. And as we've been talking about in immune system, that we have capability of generating millions of antibodies. We have capability of picking up a very diverse sequence of amino acids by our receptors. And once we respond, the responses are very specific. So if there are 3,000 different types or subspecies of a bacteria and you get infected from one, you will have an antibody to that specific strain type. So that's how specificity, and it's based upon the sequence of amino acid. So what happens is that most of the time when we are given measles vaccine or flu vaccine, so we are exposing our bodies to these antigens, and our bodies are producing antibodies. And now these antibodies are good enough to fight if there is a challenge from any of these viruses in future. And it's almost taken for guaranteed that once you have a measles vaccine, once you have a polio vaccine, you would not have the disease. So you can imagine this kind of specific response that we get from our immune system. Now, when we talk of development of B lymphocytes, originally the experiments were done in birds, and there is by cloaca, which is like a large intestine, there was some bursa, which is called bursa fabricius. And when it was discovered, that's where these cells were synthesized. They call them B cells. So basically, bursa fabricius for B cells. And human beings obviously don't have bursa fabricius, but our cells, B cells, would be made in either primary lymphoid organ like bone marrow. You can see from here, like bone marrow. And in the intrauterine life, if bone marrow is not that developed, we will have it in our fetal liver. But all you need to remember, like any white blood cells, they are going through a phase. So they are not like platelets. You have a megakaryocyte, and megakaryocyte breaks apart, and you have small little chunks of cells we call platelets, because the function of platelets is plugging. But this goes through a differentiation process. And when we say differentiation process, so we're talking about from A to Z. And anything can get arrested at any time point. So there are checkpoints over there where we may have some problems, especially for B-cell carcinomas. B-cell carcinoma. Can you tell me any B-cell carcinoma that I've talked about so far? Say it again. We talked about multiple myeloma. Remember when I talked about the antibody protein, I said multiple myeloma protein. So that is a protein, when you get excess of those antibodies produced by the B cells, you end up having a cancer, so that's not good. So everything in normal levels is good. Anything is overproduced or underproduced will cause problems. Now, this is one of the most important slides that you will see, not only in immunology, but also B-cell cancers and B-cell cancer chemotherapy and anti-cancer chemotherapy. And there will be different drugs working at different levels. So I want you to pay attention to the stages of B-cell differentiation. Again, as a rule, all cells begin from stem cell, and then we have these steps till we finally make a mature B cell. So that's like the final product. We have a mature B cell, and you can see that before we have a mature B cells, we have a pro B cell, a pre-B cell, immature cell, and a mature cell. And they are all in the blood. So many a time you must have heard in some cancer, they say, we want to take a bone marrow biopsy. So they'll take a bunch of the tissue of bone marrow and they'll run through and look at the cells. So they will then determine as to what is happening at that particular level, where is the arrest or the overproduction of those specific B cells are taking place. Now, in terms of immunology, all you need to know is that we know for sure that we've talked about the mature B cell expresses IgM and IgD. That is something that we already know. And this doesn't come right away because these are proteins, and these proteins are antibodies. These antibodies are going to act as receptors. So there is going to be a genetic differentiation or gene expression before your B cells will start making B cells, right? And then again, how do we assess them? So we look at some of these receptors. All cells have receptors, so we look at membrane expression. So if you separate the B cell and you look at the expression of B cell, that's what we look by flow cytometry. So this is something that we look at, flow cytometry, and then you will see these are antibodies, and we tag them with another antibody, and it gets fluorescence, it gets activated. Now, as you can see, there is no expression of Ig over here. As we move on, we will see expression of IgM and IgD. So if your cells are IgM positive and IgD positive, you basically are looking at mature B cell. And then there are some other markers. Remember I talked about those sequence of amino acid. Today I will introduce a term called CD, which is cluster of differentiation. So you will hear that a lot. Cluster of differentiation. So this is set of those epitopes, set of those specific markers that we discover every time we research on these cells. And they've been doing it for ages, to such an extent that now we have to come with the terminology. So they're like, there was a time when when it was first discovered, we call it CD1. Second was discovered, we call it CD2, CD3, CD4. You heard of CD4 T cells. And then so on and so forth. And now there are hundreds of them. Right? So you will have a drug in the market, it will say it's going to block anti-CD27. So that's a very specific epitope which is expressed on the brain, and that's what it blocks. So the upshot of this figure is that you can see that there is a gene arrangement of those specific heavy chains and light chains. And when we say heavy chains, how many heavy chains are there in antibodies? 2. What are their names? Kappa, no. Gamma, alpha, micro, delta, and epsilon. These are heavy chains. And what are the light chains? Kappa and lambda. So these are the light chains.
Okay, so you get IgM off the immature B cell, right? So then you convert to the mature B cell, you have IgM, and then you have the IgM.
Okay, let me come to that. Just give me some time. So what happens is that you can see when these cells are formed, there is a beginning of expression of those heavy chains. So heavy chains start getting expressed. So as we move along in maturity, the heavy chains, initially every B cell has capability of making 2 types of heavy genes. These are M and D. This is normal. So they have the ability, they will produce those antibodies, M and D, and they will get transplanted on the top of the membrane. Right? So how these antibodies are made? This is again a gene expression selection. And again, chapter 6, as I said, I'll post it on the genetics on the Moodle. But if you are interested, what happened is that there is a gene segment for antibody production, and it has got 4 portions. One is V portion, which is variable. The other portion is D portion, that stands for diversity. And there's a J portion that stands for joining. And then there's a constant portion. And there are choices for this gene to pick up a particular sequence from each of the 3. And then again, there is a whole process of transcription, splicing, translation, and protein production. And chapter 6 will talk about that, but you can see from here, once it goes through that, we will end up B cell producing IgM and IgD. Now the question will come that we may need other antibodies as well. We may need G, which is very important. We may need E. We may need D. So now we have to switch the class of antibody. This process is called class switching. So we have to manipulate, and we have to do something over here to this B cell on demand, because we don't want to produce those specific antibodies unnecessarily. We needed it because we need to place it on top of a B cell. But we don't need other antibodies at random unless there is a good reason for us to have them. Now, once there is a message to this B cell that we need to produce G, A, or E, it goes through a process where B cell will shed off M and D. They will be shed off, and they will switch to another class production, which of course will be other than M and D. So that process is called class switching. And that of course is provoked by an antigen. And antibodies are not produced by B cell alone. Sometimes they need to get help from T cells. So we need T helper cell. So this is a cell that is there to help B cell to produce other antibodies. Now, once you get exposure to a bacteria or a virus, and your body would initially want to make IgM, because IgM is something that it wants to produce as an initial primary response. Now, that IgM, of course, will come when there's a signal to this B cell that we need to switch. We call class switching. But also remember, when we discussed the other day, there's a primary response and there's a secondary response. So in the secondary response, what happens is, while B cells are dealing with a particular antigen, they are making sure that all the data for that particular antigen is stored in another type of a B cell we call memory cells. And you can see memory cells over here. Okay, so this is all the process that we basically go through. And then again, as I said earlier, you can look for a CD molecule. And if I was to look at an initial cancer, which is a cancer of a pro-B cell or pre-B cell, you can see it's going to be CD10 positive. If I want to look at the cancer for later part of B cell immature or mature, it's going to be CD20 positive. So these are some of the diagnostic tests that we use in order to pick up where in the cycle the cancer is being produced. So that basically will give you a differentiation pattern of a B cell. Now, if you're interested, let me talk a little bit about genetics, and I hope, and I've been told that many of you, how many of you have had taken genetics in the past? Ooh, that's good, quite a few of you. So, well, genetics. Now, again, I would not go in detail. I would expect that you know what is intron, extron, what is different between DNA, RNA, messenger RNA, transcription, how that process takes place. But this is a sequence of gene expression and presentation, details of which are again in chapter 6. And you can see again, in this case, what we are trying to do is that we are going to produce a heavy chain. So you want to produce heavy chain. You just told me the 2 heavy chains in an antibody, like a micro chain. Now, this heavy chain is a protein, and protein has amino acids, and we have a room to play. So we have different types of, like I gave an example of human beings. There are 5 billion human beings. They all look alike in anatomy, physiology, but if you look at their fingerprints, the detail is different. So we look at the same antibody, but if you look at the fingerprint of that antibody, the sequence of amino acids that they have on the Fab portion, that will be different. Now, I want you to pay attention to the germ DNA. Again, basic genetics, you have a 3-end and you have a 5-end. And then you have bunch of gene expression on chromosome. Now, in this case, I want you to pay attention over here. There is a leader over here that again is going to lead the sequence of events. And then there are V area, there is a D area, there's a J area. So V, D, J. These are like aggregates of the genes. And then we have those heavy chain sequences, which are either micro, or in this case, you can see probably delta. And this is like a heavy chain. If you compare that with the light chain, because light chain, we are going to pick kappa or lambda. So these are another sequence of gene. So when gene sequences happens, there is a rearrangement of DNA. So we pick up a larger pool and we want to go and shrink it up. So that's what's happening. We're going to go larger body, we want to keep on shrinking it. And you can see from here, again, 3-end and 5-end. We pick up V1 from here, and we pick up another V from here, put one of the D, and then again, you can see that we have a choice. There are 6 different choices that we have from J, but we are kind of going to compact them as we move along into the rearrangement portion. And you can see over here, VDJ segments are there. We keep on shrinking it till we reach to this level where we want to transcribe that onto an RNA. So DNA needs to be transcribed on an RNA. So we have, at least in the primary RNA transcript, you can see that we are removing those junctions between the extrons, introns. So they've been cut. So they've been sliced and things are kind of shrunk and being brought together. And you can see in the primary RNA transcript, that's what we get. We process it further. We have a messenger RNA. And in messenger RNA, we have a leader sequence. We have a VDJ. So we have 3 of these, VDJ. And then we have the fixed one, which are heavy chain. So heavy chain will form the basis of antibody. And VDJ are those numbers that we're going to shuffle around to make a very specific response in terms of antibody production. And then again, when the polypeptides are formed and the proteins are formed, you can see they will come up with a V segment, and they will come with a D segment, and then they will come with a J segment, and you will have a full-blown protein, as we say, globular protein. The only difference in this case will be, if you pay attention over here, this distal portion which says variable. So In a heavy chain, you have a heavy chain segment, FC, a bunch of smaller chain, and then finally you have the distal ends, which are hands. They are variable. So that's where one antibody will be different from the other antibody because of the sequence of amino acid that your antibodies have picked from those choices that it has over here. So you can see it has tons of choices. For example, in D, it has about 23 choices. In J, it had 6. So it's like mix and match. It's going to pick a totally different sequence of amino acids. And then in a variable portion, you will have the distal segment heavy chain, and then the small chain, light chain will come over here, and then you assemble an antibody. So, of course, in chapter 6 talks about the enzymes. We are also interested in enzyme. There is one enzyme which is called VDJ recombinase. That is an enzyme that will sort it out. It's going to pick up 1V, 2D, whatever different. This is the major enzyme that we want to work with, and there are quite a few drugs or even bacteria or viruses that will kind of go and affect that. Now, just to summarize, we talked about the early stages of B cell differentiation. We talked about a pro-B cell, and then you can see there are different CD markers expressed. Then we have pre-B cell. You don't really have to remember all these things, but remember when we say tissue typing, when we say bone marrow biopsy, then you wonder what is it? What is it they're going to do? What are they looking for? They're looking for those cells. They'll pick up those cells. They look at the external markers they have, and they give you a sequence of events, especially at genetic level. So that's what's happening over here. So VDJ for the heavy chain. You can see H stands for heavy chain, and micro, of course, because most of them start with the micro, because micro is the basic structure of the heavy chain. Now, another important thing you have to keep in mind that the basic theme of immune system is that you want to make sure it differentiates cell from non-cell. Just imagine for a while that your B cells are making these antibodies, and all of a sudden something weird happens in these receptors, because these are receptors in the end, and we call receptor editing. So they edit a receptor based upon your genes. That particular receptor is the same that is present on your cells. So what will happen now? That this antibody is designed to react to a sequence of amino acid, and your cells also have that. So that will be a big problem. And that happens. I gave you an example for SLE, rheumatoid arthritis. Some of them, people think diabetes mellitus, have a genetic immunological link. We don't want that to happen. Now, per chance, if your body in this kind of random sampling picks up a sequence of events, it's just like 100 of you, you're talking to each other and you say, what's your cell number? They said, my cell number is the same. What are the chances? None, right? I mean, practically none. So this normally does not happen, but in the world of when you're working 10 to the 15, it might happen, and it does happen. So in this case, what do you want to do? If you make those kinds of antibody, you want to make sure that you delete those antibodies. If you make a mistake, right? So if such a thing happens, you happen to have the same number, you want to call those people up, they want to delete information on one. Exactly that's what our body does. And the process that our body does it, and you know that, you already taken pathophysiology, is called programmed cell death. What is the other name for programmed cell death? Apoptosis. I've seen people in scientific conference saying apoptosis or apoptosis, whichever way you want to go. So apoptosis, we don't want that. So this means, as a matter of fact, very interestingly, each and every cell in our body is programmed. So we have a program that this cell, after the stem cell, will become a liver cell. a kidney cell, a B cell. So there's a program there in the gene. And also there is a program which is just like a product that you make. You say manufactured such and such time, and expiry date as well. So this has an expiry date as well, right? So exactly all cells in the human body are programmed. And what our body does it, that when there is a problem with VDJ rearrangement, right, and it is transcribed or translated into a good microchain, it will keep it because it needs it. If it makes mistake, so there is an assembly line. So there is an assembly line, Ford factory, car factory. So there are people who are going to inspect at each and every checkpoint. If something goes wrong, the chances are they're going to send it back for repair or whatever they want to do. They will give at least one more chance. Exactly, that's what the body does. It doesn't want to ruin your effort. Okay, you did something. Let me give you another chance. It's not like micro, we don't give you any other chance. But they give you a chance, so they'll give you a retake, another chance to fix the problem. But if in the second chance, the problem is not fixed, so that chain, heavy chain, cannot go. So what's happening in this case is that this VDJ is basically coming from your parental chromosome. So you get half genetic material from your father, half. We look at phenotypic expression of our parental genes that are in our expression, eye color, hair color, skin, so on and so forth. But the fact is, it's on the gene level as well. So that's where they will pick up, and as I said, that they will give them a second chance, and if still they are not rectified, the cell will die by apoptosis. Now, I talked about receptors, and remember, receptors, very important. We had receptor physiology, we talk of receptors everywhere, because just like acetylcholine receptors, just like autonomic, so there are tons of drugs. They are either going to upregulate, downregulate, do whatever they want to do, because receptors are gates. You have calcium channel blockers, right? So drugs apply. So there are drugs that specifically talk of receptors, and we need to know the detail for that. So in this case, when we talk of pre-B cell receptor, basically what happens is that B cell has to take a signal. Remember, B cell has to talk to an antigen. Now, B cell as a whole has two receptors there. One receptor we call simple B cell receptor, BCR. So this is the one that is going to talk to the antigen. Or we may have a pre-B cell receptor. Pre-B cell receptors are immature, and B cell receptors are mature. And these are happening in your bone marrow. So it may happen that because of some reason, cancers, the fully matured B cell do not come into circulation, and you have a premature or pre-B cell that comes over here. So it's going to be non-functional, or it will do weird thing. Now, as a rule, what normally happens is that, for example, signal, just one signal, you give a signal to a B cell which has IgM and IgD that, hey guy, we don't want M and D, we need to produce other antibodies. So what exactly is the signal? That's what we need to understand. That's called signaling pathway. Does that make sense? So in this case, what happens is, the series of events from A to Z where we transmit a signal. So transmission of signal in immunology is called signal transduction. Just like those of you who know electronics, and they are working with many gadgets over there. It's basically a transducer. It's like a transducer. Now, a mature cell will bind an antigen, through those receptors and send a signal to your B cell. Let me give you an example of a vaccine. For example, some of the vaccines are antibodies, passive vaccines, some of them are antigens. Flu vaccine. So what they do is they pick up the antigen that will not cause a disease, give it to you. So this antigen goes into your blood, goes into the lymph node. There's a B cell sitting over there. B cell will capture that antigen, and then it wants to send a signal. What do you want to do with this antigen? Now, antigens are known to be responded in terms of antibody production. So you'd say, well, this is so-and-so, 6, 7, 8, 9. They give a strain number. There's a sequence number of amino acid. Say, we want you to make antibodies against 3741, for example. So this signal is transduced from exterior to inside. And this is a series of steps, right? And all these steps have enzymes. All these steps have molecules. And we have at least hundreds of drugs acting on each and every step. And that is the basis of HIV therapy, that's the basis of cancer therapy, that's the basis for those of you who are interested in working in R&D and want to develop a drug. That's a drug development thing. So you need to understand what is it that is going on. Now, let me talk about a clinical example. So keep that in mind. One of the problems that we have is called Bruton's tyrosine kinase. So they are, and I'm pretty sure that you know more biochemistry than me. So you know there are tyrosine kinases, there are phosphokinases, and there are a bunch of those other enzymes that basically carry this signal transduction, right? And there are motives, there are inducers, so on and so forth. Now, one of the enzyme that is involved in this type of signaling, if it is absent, that happens in some of the male children, we call it Bruton's disease. We'll discuss that in detail when I talk of immunodeficiencies. So immunodeficiencies are those diseases when you don't have that proper production of antibody, and they are other diseases we call X-linked A-gamma globulinemia. So what do you understand by A-gamma globulinemia? Medical terminology word. What exactly does it mean? A-gamma globulinemia. A means absent. Gamma globulin is an antibody. Anemia means in the blood. There are no antibodies in the blood. Correct? Let's see. What is a receptor? So when we say receptor, we want to figure out what exactly is a receptor. Now, this compares a pre-B receptor versus a B cell receptor. Now, remember the pre-B receptor has many different things. I don't want you to remember all the details, but I would rather pay attention to actual B cell receptor, and you're familiar with that. There are heavy chains, and there are light chains, and there are disulfide bonds over here, and then these are the sites for antigen binding. Now, I gave you a very crude mechanism the other day. The antigen binds to an antibody, antibodies enter cytose, breaks apart. That may happen. But to be more specific, in terms of signal transduction, in terms of something that transfers the signal, so you can see when an antigen binds over here, there is something happens, the message goes, and this is a transmembrane, this is a membrane over here, and then again, this is attached to one of the other molecules that I discussed yesterday, Ig superfamily. I want you to pay attention over here that you will see the BCR has a micro chain, which is a heavy chain, but of course it could be gamma, alpha, so on and so forth. Now, there is another structure that sits next to it, and we call it ITAM. These are like ITAM. ITAM stands for immunoreceptor tyrosine based activation motif. So these are ITAMs. So these are like ITAMs which are depicted, and the moment it's just like you turn the switch on and the electricity follows. The moment an antigen comes and binds to it, there's a series of events we call signal transduction. One of them is basically activation of item, and I'm going to discuss that in very detail. And when I talk of signal transduction, what actually happens. So this is this membrane event, signal comes over there, these are two polypeptides and they will come closer and the signal goes. Right now, just remember that there is a immunoreceptor, tyrosine, and the word suggests tyrosine. So this means if this is a tyrosine-based activation motif, you will have to have tyrosine kinases within a cell. They will come and dock onto those specific sites over there and transmit the signal. Okay, now responses. And let me give you, I think it's better for you to have a visual and then you can read this on your own, because sometimes you learn in a visual way better, because otherwise it becomes very difficult. Now, we've been talking about B cell being exposed to an antigen. So far, that's what the theme was. Now, what happens? Now, in this case, this basically gives you an example what may happen if you have an immature B cell. Now, you wonder, the chances are the immature B cell may respond to your self-antigens. If your body is making immature B cell, it may respond, it's going to go everywhere and start chewing you up because of the problems. Because some of the diseases are such, they are systemic diseases where you make these kind of autoantibodies, that's what we call them, and they will pick and destroy you. So in order for us to treat them, we have to remove them, or we have to do something that needs to be done. Now, antigens can be presented on the cell-bound antigen, like a self-antigen. If it is in bone marrow, so there are cells in the bone marrow, B cell is produced, it's an immature B cell, or there's a soluble antigen. This is happening in bone marrow. Now, what are the choices that your body has to deal with this kind of a situation, because it's in bone marrow? So what happens is, number one is when it sees an antigen, we talked about B cell will code it. It's going to go and punch the numbers, so to speak. We call it receptor editing. We want to find out this number, and we want to make sure that we make those type of cells circulating in the body that do not have self-antigen. Because in bone marrow, when the B cells are produced, if you let go this B cell into circulation, then of course they will circulate. Within 12 hours, they would have done damage, because it takes only 12 hours for a newly born, naive B cell to circulate and cause problems. Okay. Now, if, for example, it does not pick up self-antigen, it's good to go. So we'll let it go. If, for other hand, if it picks up and responds to self-antigen, we want to remove it by apoptosis. So that's another mechanism. But these type of mechanisms that we have is only for self-bound antigen. But there is another spin to this process where you may ask yourself, what happens when an antigen is a soluble antigen? It comes in contact with a immature B cell. So we have another process. We call energy. And then again, I would want you to remember that. See, some of the terms, like a bibliography, if you see at the end of the book, you will see definitions of these terms, ITAM, CD4, all BCR, TCR. These are like the language, the French that you may want to learn to understand immunology. But what happens is energy means long-term inactivation. So we energize the cells. And that's a process sometimes we want to use for cancer cells. The cancer cells are malignant. They are causing problem. We just want to cool them down. We just want to remove the whatever markers are there or that are causing damage. We want to energize, not with E, but A is opposite of energy. So you can see is an inactivated B cell, so it's like useless, so to speak. Okay. Now, the interaction of an antigen with an antigen specific, if those of you who remember when we talked about B cell and T cell, I talked about what type of antigens are there. There were protein antigen, carbohydrate antigen, and so on and so forth. So far, you may have got this impression that B cell takes care of all the antigen on its own, which is partly true. But remember that especially if there is a protein antigen, there is always an input of a T cell. So when it says T cell, now we would divide antigens and antigen processing into two different categories. One we will call thymus dependent, which are mostly proteins, and one we will call thymus independent. They are mostly non-proteins. Now in this case, as you can see from here, the idea is that thymus independent, TI, do not need T cell help. The classical example is polysaccharides, a bacterial capsule. So also keep in mind some of the vaccine like HIB, they are made from the polysaccharide of the bacterial capsule. They are like good vaccines because they will induce your immune system to produce antibodies without help from T cell. So let's see what happens when an antigen comes. This is a lymph node, and you can see lymph node has germinal center. It has places where there is abundance of B cell and T cell. Now, this is one antigen. So antigen can come and circulate into lymph node, and there is a B cell with those receptors. This will interact. So that's the first interaction any antigen may have, especially the one which are circulating. The other other route for the antigen is that they can be grabbed by a dendritic cell from the skin, mucosa, everywhere. They'll grab those antigen, pick it up, and bring it for processing into lymph node. And you can see especially over here. So this is like a T cell zone. A lot of T cells, this is a B cell zone. But they need to talk to each other. Exactly, that's what we say. When the cells communicate with each other through receptor, we call talking. So cells talk to each other, right? So this communication, of course, is important, especially when you have protein antigens, right? So you can see from here, let's say if there is no protein antigen, so it's not thymus dependent response, you don't need T cells. So what will happen is the message will come to a B cell, it will start making antibodies. Right? But I also told you most of the time the antibodies that these cells basically will make are antigen specific. So there is going to be an antigen specific response. And what do you think will be the first antibody that these B cells will produce after they have seen an antigen? IgM, right? So IgM. So you can see from here, early synthesis of IgM followed by some B cells and few T cells, then they can basically migrate back. So this actually is happening. Remember, I also told you sometimes you have a cut over here or injury over here and the lymph nodes are swollen, right? Because of the production of these things that are happening. Now, antibody synthesis per se, as I said earlier, if it's thymus dependent, that's happening in germinal centers of lymph node. And typically, like I just explained earlier as well, that B cells will pick up a gene from VDJ region. And if there is a message for the cells to switch from M or D to make any other thing. There's a class switching that will take place again. And then again, the B cells will make sure that they always keep a memory cell. So every antigen that enters at that level, you always keep that information reserved. And this memory cell basically do not express any antibody on top of that. That was another thing. And they are long-lived cells. So one of the purposes of vaccination is that a good vaccine will make a good memory cell in your body. So whenever you get that challenge again from that particular microbe, so memory cell is sitting over there, it can recognize that, and it knows the sequence, it start making antibodies that are required. Okay, now you can see from here, this is a lymph node, and these are like upshot of the events that I've talked so far. How many of you have heard about cloning, right? You must have heard about cloning, right? Cloning, right? Cloning simply is copying. So you can clone a copy, copy, paste, tons of them. There's no limit to that. So the B cells, once they pick up this information in the beginning, then they can expand, clone. And the whole idea is that they make similar type of cells, so you can see tons of them. So there's the outer mantle, there's a dark zone, and there's a light zone, and you can see that once an antigen comes, and the antigen is seen by a... Why does antigen see B cell first and not the T cell first? There are more T cells. Anybody else? What are the cells that have the capability to process antigens? B cells, not T cells. So the cells that have capability of processing an antigen, pick it up and process an antigen is dendritic cell, cells of monocytic or macrophage cell line, and B cell. So these are the classical antigen presenting cell. That's where they are sitting everywhere. Okay? So they will present an antigen, then of course they can seek for T cell help, and then they will make what they... There was another thing they are producing is our soluble protein cytokines. There's a full lecture on that. And then again, a bunch of drugs that we have on cytokines. So these are some of those molecules that are produced when this activation takes place. Now, also remember that some of the antibodies that we want to stay in the circulation will be made in lymph node, because lymph node is in direct contact with lymphatic system, and lymphatic system is circulating with the blood. So there is a closed system where you can get this antibody circulation. But remember, when we talk of IgA, for example, IgA is basically expressed outside over here. So this is a gut-associated lymph node. You can see GALT. GALT stands for gut-associated lymphatic tissue. And MALT means mucosa-associated lymphatic tissue. These are some of the acronyms that you'll find in the book. So now the question is, there are tons of bacteria, viruses in our gut. They're always there. There's nothing you can do. So they are sampled by Peyer's patches. We have M cell. They sample them, and they induce production of B cell. So now these B cells basically know that we have to produce IgA. So they are committed to form IgA. They'll make IgA, but they will put a tag on there. So they'll put a tag on there, and this tag we call another concept homing. The idea of homing is that any cell or an antibody, once it's tagged with homing receptor, it will always home back to site of induction. So this means if this was the home of that response, the cell will be committed, made, and then goes back to where it was induced. So in this case, what is happening again is that because we don't need IgA in blood, we need IgA on the mucous membrane. And remember, I told you that IgA has to travel across the epithelium. And I also told you that IgA needs chaperone. So you need very specific receptors. So you can see from here, this is a poly-Ig receptor. And once we have IgA antibody, it will attach over here and secrete it as a dimer. So this IgA basically will sit as a part of mucous membrane on top of your cell. Okay? And final slide. Okay, I'll stop here because I think I see that